So here's the exciting thing. There's a lot of steps and a lot of process. Ooh, sorry, guys. Sorry. I got to kill time while these warm up. All right. Anyways, there's a lot of steps to actually facilitate a muscular contraction. Okay. And we're pretty much going to pick up where we left off with the neuron. All right, we saw the action potential rushing down the axon, going down to the synaptic knobs, and then releasing that neurotransmitter into the synaptic cleft, goes into the cleft, and then it's going to do something to the effector organ or the other neuron. So that's what we're going to talk about now. Now we're going to be talking about the effector organ, and in this case, it's skeletal muscle. All right, so if we're dealing with skeletal muscle, that means that neuron comes from the somatic nervous system. That's the part of the nervous system that we can voluntarily control. All right, so there's going to be 3 different processes that we'll talk about. We're going to talk about what happens at the neuromuscular junction. Okay, we've kind of talked about that in chapter 12. So some of this will be reviewed for you, so that'll be good. Okay, and then we're going to talk about what happens at the plasma membrane or the sarcolemma of the muscle cell, right, as that action potential. Because remember, we got to generate an action potential, and it's got to find T-Tubules. Remember the T-Tubules? The T-Tubules are the invaginations. They're those holes, the well, like a well goes into the ground. All right, the T-Tubules are the well that goes down into the skeletal muscle fiber. And that action potential will go down in there, and it's going to try to open up the calcium gates, all right, the voltage-sensitive calcium channels, all right, because remember, we got to release calcium. It's trapped. We need that calcium to escape, to get out there and flourish, right, into the sarcoplasm, because it's trapped right now in the sarcoplasmic reticulum, these purple tubes, and also here in the terminal cisterna. The ends of all these tubes here, terminal cisterna, sit on either side of a T-tubule. Okay? So then what's going to happen is we're going to learn what happens when calcium then enters into the sarcoplasm and how it's going to facilitate what we call the cross-bridge mechanism and how we're actually going to get a muscle fiber to contract. So we're going to go step by step through all of these. Okay? So we're going to start off here with the neuromuscular junction. So we're going to talk about what happens right here. All right? So let's do a quick review. Okay? All right, so the neuromuscular junction, and if you dissect the word neuromuscular, it tells you what exactly is going on. This is where the neuron, all right, the presynaptic neuron, in this case, right, is going to hover above the skeletal muscle. Okay? So this neuron is going to, hold on one second. Just keep writing. It's not too bad. You guys deal with that. It's kind of cut off on the left side there. I'm sorry. Just a little bit. I don't know why it's like that. So anyways, so we're going to see, all right, we're going to deal with the neuron, all right? So it's going to include a neuron, the cleft, the synaptic cleft, and then the actual muscle tissue, okay? And that area of the muscle fiber that the synaptic knob kind of hovers above is called the motor end plate. So there's 3 components when we're talking about a neuromuscular junction. The synaptic knob, the motor end plate, and the synaptic cleft, the gap in between those 2 tissues, or not tissues, but those 2 structures. All right. Normally we'll see this, all right, in the middle of the muscle fiber. Okay? Because when that action potential hits, all right, or that signal hits the muscle fiber, it's like throwing that rock. We use that same analogy, but for a different concept. Throwing a rock into a puddle, all right, or a lake. It just spreads out in all directions, all right? So it's better suited to be in the mid region there. Okay? All right, so we're familiar with the synaptic knob. Okay? That's where our synaptic vesicles are. Okay, those are those little tiny bubbles that are filled with the neurotransmitter, but now I get to use, instead of saying the generic term neurotransmitter, now I get to use the specific type of neurotransmitter, acetylcholine, ACH. Okay? So that's the neurotransmitter that are going to be in these synaptic vesicles. All right? So meanwhile, we have our calcium ion pumps are working day and night, all the time. And they're pumping out calcium. They're keeping it out of the synaptic knob. So therefore, they are creating that concentration gradient. Concentration gradient, again, is an area where there's a high concentration of something and a low concentration of something, a difference in concentration of chemicals, substances, okay? And so, when that action potential comes down, all right, that synaptic knob from the axon, it's going to stimulate the opening of these guys, okay? It's going to stimulate the opening of the voltage-gated calcium channels. And when that occurs, calcium enters into the cell by the process of diffusion, because it diffuses down its concentration gradient outside the cell, higher area of concentration. So it diffuses from an area of high concentration to an area of low concentration inside the cell. Those calcium molecules will then move towards those synaptic vesicles and attach themselves onto the synaptic vesicles because they have specialized proteins on them that help to bind the calcium. That calcium is positively charged. And you guys remember that the inside of the plasma membrane is negatively charged. So opposites attract. So those now positively charged synaptic vesicles get drawn towards the electrical negativity charge of the inside of the plasma membrane near the synaptic cleft. So they migrate towards that direction. They fuse with the plasma membrane through the process of exocytosis. They release their contents, in this case, acetylcholine, into the synaptic cleft. And then that acetylcholine diffuses across the synaptic cleft to the motor end plate on the other side. Okay? So on average, we'll release about 300 vesicles. And those 300 vesicles from the synaptic knob contain thousands of acetylcholine molecules. That's a lot. So that's just going to be floating around in that synaptic cleft. Okay? So that's what happens here at the neuromuscular junction. Not bad, right? We know that. We feel pretty good. I like it. So I'll walk you through it on a picture here. All right. Here you can see, here comes that action potential flying down the axon. Finds its first voltage-gated calcium channel. Calcium's in high concentration outside the cell. Calcium enters into the cell. It attaches itself to these specialized proteins all over these synaptic vesicles. Okay, the inside of the plasma cell, the plasma membrane is negative. So these positively charged calcium ions get drawn towards the synaptic cleft. All right, these synaptic vesicles fuse, okay, with the plasma membrane and release acetylcholine into the synaptic cleft here. On the other side, we have our acetylcholine receptors, okay? And so that acetylcholine then will attach onto those receptors and it will trigger a magical process. Okay, we're going to create, the beginnings, not an action potential yet, but what we call an end plate potential. So we're going to talk about that. But that comes in the second phase. So that's the first phase, all of that. So this question here asks, what triggers the binding of synaptic vesicles to the synaptic knob membrane to cause exocytosis? Okay, the action potential triggers the opening of those calcium gates. Okay, so calcium will then bind to those synaptic vesicles, and that is the trigger. So now you can see calcium is important, right? At least so far. we learned from our video last week that we need calcium to remove troponin and tropomyosin off the binding sites for actin and myosin to have a cross bridge. I'll show you that video again here shortly. Okay, but now we can see why calcium is also important. Neuron transmission. Okay, so now we're going to move to, all right, that was the first part. Here was our first part. Okay, neuromuscular junction. Okay, now we're going to see how we're going to cause excitation here at the motor end plate. Okay, all right. So keep in mind, our eventual goal is going to be to get the filaments, actin and myosin, to slide past each other. All right, but first we have to generate what's called an end plate potential. End plate potential occurs in the end plates. What's the end plate? That is the area on the other side of our synaptic cleft. All right, on that muscle there. Okay. So we call this excitation contraction coupling. All right, starts off with the end plate potential. All right, then we're going to generate what we call the muscle action potential. All right, and eventually that muscle action potential was going to travel into those T-tubules, and it's going to stimulate the release of calcium from the terminal cisterna and the sarcoplasmic reticulum. So we have to release, remember, there's our princess. Calcium is held hostage, not hostage, prisoner. All right, calcium needs to get into the sarcoplasm, okay? But it can't, because right now it's being sequestered inside of the sarcoplasmic reticulum. Okay, so that's our goal. That's our mission. So this picture here, I'm going to zoom in on it. That's what we're seeing here now. Okay, we're seeing all this neurotransmitter, the acetylcholine, is stimulating the opening, all right, of these gates here. Okay, and because of that, we're going to see same type of situation. Sodium will enter into the cell. Potassium will exit the cell. Okay? We need to get a threshold value because we have a membrane potential going on here. All right, in the neuron, our membrane potential was what? What was the resting membrane potential? What's the value? Negative 70. That's right, negative 70. You remember what the threshold value was? Negative 55. Okay, good. So these are close, but they're not the same. Okay, in our muscle, all right, the end plate potential, all right, negative 70, excuse me, negative 90. We need to reach negative 65 millivolts to initiate an action potential. Oh, excuse me, a muscular action potential. Let me be specific on that. Okay, so we've got to go from our resting membrane potential, that negative 90, all right, to the threshold value of negative 65. Okay, so that's a difference of 25 millivolts. Okay, before it was only a difference of 15, negative 70 to negative 55. Here we're going from negative 90 to negative 65. Okay, all right. Again, we need to hit a certain value here. All right, so what we're going to see is, all right, we're going to try to create at the motor end plate. All right, we've got to try to get to that threshold value, all right, because once we get, all right, away from that motor end plate and out to the sarcolemma, it's the same type of scenario that we saw in the axon, okay? Once we open up one of those voltage-gated sodium channels, we knock down the first domino, and they're all just going to go falling down, okay? But we need to get to this value here. So we know that concept, okay? And we also need to find a T-tubule. More than one is better, okay? But we got to find T-tubules. Remember, they're scattered all over the place. They're everywhere, okay? So that's what we're going to go through here, all right? So you can see, whoops, hold on, no. Okay, so we're eventually going to generate, all right, our muscle action potential, okay? And it's going to go along the plasma membrane, down the plasma membrane, okay? And it's going to find a T-tubule. Okay, it's going to travel down to the T-tubule until it encounters these voltage-sensitive calcium channels that will trigger the opening of the calcium release channels. Calcium release channels are these channels that are located, all right, on the terminal cisterna. The terminal cisterna are on the ends of the sarcoplasmic reticulum. That's where your calcium is stored. Once that happens, calcium will flow out of the sarcoplasmic reticulum into the sarcoplasm. That's where the actin and myosin are. That's where our sarcomeres are. That's where we're going to get the cross bridging there. Okay? I'm going to show you that video again here really soon. All right, so let's just kind of begin here at the beginning of what the heck an end plate potential is. Okay? So we just released our acetylcholine. It's diffused across the plasma membrane. It's attached itself to the acetylcholine receptors. Okay? And that's going to trigger, guess what? Chemically gated channels. What's the chemical? What's the chemical? Acetylcholine. That's right here. Bingo. Acetylcholine. That's our chemical. That's going to open up the chemically gated channels. All right, we know this story. It's been told 100 times by me. Well, in my career, many more than that. But to you folks, it's been told a lot of times. Okay? So sodium enters in. All right, and potassium will leave. Okay? But sodium rushes in. And what's going to happen? That sodium is positively charged. It's going to make the inside of the cell, all right, more positive, less negative, okay? And where this occurs, that's called the EPP, or the end plate potential. So it's, I don't want you to confuse the two, all right? It's similar to a graded potential because it's local, all right? An EPP only occurs in the end plate, in the motor end plates, all right? It's local, okay? So what we'll see here, all right, because it's localized, it's not going to travel far. We just have to find a voltage-gated ion channel, all right, outside of that end plate, all right? So these voltage-gated ion channels are going to be in the vicinity. They're going to be adjacent or close to the motor end plate. All right, but they're found on the sarcolemma. What's the sarcolemma? The plasma membrane of the muscle cell. Okay? All right, so we have to, again, what is our threshold value? -65. All right, so we need to make sure, because again, we're dealing with a local signal. So a local signal, all right, it has the potential to die off or lose intensity. Okay, like our graded potentials did. So we have to generate enough of them or a strong enough end plate potential to reach our threshold value of -65. All right, questions about that? Do you folks kind of see that? Similar to the graded potential, okay? Remember, our threshold value is -65. We're starting at -90. So when sodium rushes in, it's going to start to make the inside of the cell more positive. It might make it go down to -87, but that's not threshold value. Might make it to -72. Still not threshold value. We got to get to that -65. How do we do that? We open more gates. We open more chemically gated channels. We get more acetylcholine being released. We can get more sodium to enter into the cell. More sodium, more positive charge, the more that we can drop our membrane potential. We can go from -90 to -70. Oh, we're close. Let's open some more sodium gates. Boom, now we hit -65. Okay, see where I'm going with this? Saw how it was with graded potential before. Okay. Okay, so again, our goal is to get to that threshold value. Once that happens, then guess what? Bang, we got our action potential. Okay, so how do we get an action potential? We have to depolarize that cell. Make the inside of that cell, all right, more positive. Okay, so here's how it happens. Okay, the EPP reaches that threshold value of -65. And there's a nearby or adjacent voltage-gated sodium channel. Once it opens up, it's game on. That first domino falls down, the cell depolarizes, and it's going to go through that same scenario. All right, the inside of the cell will become a +30 millivolt value. I'm going to come right back to that. So that's what we're going to see on this graph here. Here's one of those graphs again. All right, so we get depolarization of the cell. It shoots up here to that positive 30. We saw that in the neuron. All right, when the voltage-gated sodium ion channels open up, okay, they depolarize the cell and it goes from a negative value on the inside to a positive value. Okay? And so that whole scenario will occur all the way down the sarcolemma, like we saw in the axiolemma. Okay? So the adjacent voltage-gated sodium channel, that will open up. And then the next one, and then the next one, and then the next one. All those dominoes will fall. Okay? It's a chain reaction. It goes all the way down. And it looks for these guys, the T-Tubules. T-Tubules, like I said, they're everywhere. You know? It's like if you were to walk into this room and you saw 100 holes just randomly dug in the floor here. Those are the T-Tubules. Okay? Those are the T-Tubules. And that's what we're going to see here. They're all over the place. Because you saw here, just to remind you, in our T-tubule, I'll come back to that slide, all right, in our T-tubule, all right, that's where we got to get our action potential to go down into, because we have to stimulate the opening, all right, of these voltage-sensitive calcium channels, all right, because they'll trigger the opening of the calcium release channels in the sarcoplasmic reticulum to release calcium. Okay, that's what we're looking for. All right, so our goal, always, never lose sight, is to release calcium into the sarcoplasm, all right? But to do so, we got to get our action potential to head towards this T-tubule. Not just one, but many. The more the merrier, okay? Because the more sarcomeres that we can get to contract, the more, if we need to have a forceful muscle contraction, we can do that, all right? So we need to release more calcium. All right, so far so good, not too bad, similar concept. If you understood the neuron stuff, okay. All right, then this kind of helps. Now you can see why we teach chapter 12 first, then do chapter 10. Okay, similar type of scenario. Okay, so same type of situation that we're going to see here in the muscle as we saw in the neuron. Following, all right, the opening of the sodium channels, all right, we are going to see the opening of the potassium channels as the sodium channels close. Okay, so now it's potassium's turn to do something. So guess what? It leaves the inside of the cell. It takes its positive charge with it, and it causes the cell to repolarize. And it gets us back down to that starting value of negative 90 millivolts. Okay? And the repolarization occurs the same way that depolarization did. All right? It spreads out all along the sarcolemma. Okay? And down into the T-tubules. All right? So a cell will first depolarize, then repolarize. Okay? So like the neuron, we had a refractory period that followed the action potential. All right? We see here in a muscle cell, we've got another refractory period. Okay? And that's that period of time where we cannot, well, we'll talk about exactly, because the refractory period is going to be a little bit different in the muscle than it was in the neuron here, all right? But we are not able to stimulate, all right, or respond until another stimulation occurs. Okay? Sorry, I got messy. All right, so there is a figure, I think it's figure 10, 12. In your book, basically what I just explained to you, all right, in pictures and whatnot, will be explained on this diagram. And it shows that the action potential, the millivolt value, and of depolarization versus repolarization. Okay, so you can see here, all right, again, what I would say is learn this here. If you feel pretty comfortable with this, then you can move out to the more detailed explanation here. Okay? So we start off with our resting membrane potential of negative 90 millivolts, okay? And then we see as acetylcholine is released, all right, and it triggers the opening of those chemically gated ion channels, okay? Sodium enters into the cell, all right, and it causes the inside of the cell to become more positive. So our resting membrane potential goes from -90, and once it hits -65, that's our threshold value. Then we get depolarization, but that occurs at the voltage-gated sodium channels. All right, and that is going to be located in the sarcolemma. Okay, it's not in the motor end plate. All right, chemically gated channels are in the motor end plate. All right, our voltage gated channels are going to be in the sarcolemma. Okay, so once we open up the first one, bang, it gets propagated just like an action potential in the axon. All right, and it's going to go down the entire length of the sarcolemma down into T-tubules. All right, so that will bring our value from -65 to +30. That's here. The upswing, that's our depolarization. Okay. Once we get to the +30, the voltage-gated sodium channels start to close. Voltage-gated potassium channels start to open up. Potassium leaves the cell. Here we have repolarization. And repolarization follows the depolarization all the way down the sarcolemma into the T-tubule. So we return our value back to that -90. Okay, we'll dip a little bit below it. Okay, no big deal. Okay, we'll quickly fix that and get back to our -90 value. Okay, a little bit easier with muscle. Depolarization, repolarization. This is an important concept for you to know because you're going to see this again when you get into the heart, when you talk about cardiac muscle. So I really want to hammer that home. Depolarization, repolarization. Okay? Two things follow depolarization. Repolarization and a contraction. Right? That's it. Okay? All right. So we're almost there. Okay? We're almost done with the second phase. Okay? So our action potential has now traveled along the sarcolemma, and it finally makes it to a T-tubule, hopefully many. Okay? And it travels down the T-tubule until it finds those voltage-sensitive calcium channels. And they will then cause the opening of the calcium release channels. And just like the name of the channel says, calcium release, it releases calcium, where it was held captive in the sarcoplasmic reticulum terminal cisterna, and it diffuses out of the terminal cisterna into, yay, the sarcoplasm. And this is what we've been waiting for. This is what our goal was to do this whole time, is that we wanted calcium to interact with actin and myosin, not really actin and myosin, but troponin and tropomyosin. So we can trigger a muscle contraction. If I could make fireworks appear on this slide, I would. All right? That's the goal. That's what we want. That's what we're trying to do. Okay? This is what we've been trying to do this whole time, is get calcium to be released into the sarcoplasm. So we can get actin and myosin to link up with each other. Hooking up. It's like a matchmaker. Calcium is our matchmaker. Matchmaker, matchmaker, make me a match. Does anyone know what that's from? Thank you very much. Sung by which character? Do you remember? Tevye? No, not Tevye. Lazar Wolf.
Yeah.
No, was it Lazar Wolf? The Butcher.
The Butcher, I think, yeah.
Yeah. Anyways, I only know that because two of my best friends were in our production of Fiddler on the Roof in high school. Okay. Not too bad so far? Not too bad? Okay. Let's tell you what. Let's put this into, okay, a picture, a video, I mean, okay? And let's see if this video hopefully makes a little bit more sense. A little bit more sense. I'm thinking it might, okay? I'm thinking it might. Okay. Here we go.
You use muscle.
Hold on, lady. Oh, are you kidding me? Phew. All right. All right. For you at home, give me one moment. We will have you see what's going on here. All right. So we're going to show that video here. At least I can control that from the TV screen here. Okay. Let's watch this video and see if it makes a little bit more sense to us. Okay. Here we go.
Muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril.
All right, so you can see here, this is showing us that's the contractile unit, all right, and it goes from one Z disc or Z line to another Z disc or Z line. We're going to start talking. We talked about this last week, okay? Actin and myosin. Remember the M line, H zones, I zone, all that. Okay. So we're going to kind of review that a little bit here, but we're going to really get to the other important stuff that we're going to hit up really quick here.
Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filament...
So you can see here where there's some overlap between the thick and the thin, all right? And that's going to appear darker. And then you can see where there's just thin, that's going to appear lighter. Now you can see why we have striations in skeletal muscle, all right? It's because of where there's overlap. Where there's more overlap, those areas will appear darker.
Filaments are anchored to the Z lines. The sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate.
You haven't talked about this, but we will sometime today, okay? So you're going to understand this, so don't worry. It's the part that comes after this that you'll understand.
This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax.
So you see why you need ATP? It's the ATP that allows the myosin head to disengage from the actin. All right? Now, maybe prior to that you thought it was the ATP that caused the power stroke. All right? A lot of people think that because you just assume that's energy, an energy using requirement. Okay? But it's actually the ATP is needed for the myosin head to disengage, all right, from the actin to either recock and do another power stroke or just to relax. Okay? So keep that in mind. Here's why calcium is important. Here we go.
Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin which displaces tropomyosin, exposing the myosin binding sites on actin.
Boom. That's what we're just trying to do. This whole time what I was trying to explain to you comes to that. We needed calcium to do that. All right? Our whole job with that whole neuromuscular junction issue and the action potential propagating down, all right, the sarcolemma down in the T-Tubules was to get to here, to get calcium out of the sarcoplasmic reticulum into the sarcoplasm, because that's where the sarcomeres are. That's where the actin and myosin is going to be, so we can get the calcium here to do this.
This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T-tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Do you understand it a little bit more? Did that video make a little bit more sense to you, at least, after what we talked about? Okay, cool. Now, again, there's the other part with the binding and the cross-bridging. We'll get to that, okay? We're going to get to that. Okay, so let's get back into our safe zone, hopefully. All right, and then we will now move on and talk about, because finally we've got calcium to be released, so now we're going to talk about that cross-bridging. Where was I? Right here. Okay, so question. What two events are linked in the physiological process called excitation, contraction coupling? I can't talk tonight, okay? So the two processes that are going to be linked are going to be, all right, what happens at the neuromuscular junction, all right, and our sliding filaments. So again, in the middle, all right, we have that excitation contraction coupling, okay? So we have to, there's those two parts at either end. What occurs at the neuron and the motor end plate, and then we have the sliding filaments, all right, but in the middle, that's where we have our excitation contraction coupling, okay? What we just talked about, okay? That action potential rolling down the sarcolemma, all right, heading towards the T-tubule, releasing calcium from the sarcoplasmic reticulum terminal cisternae. Okay, so the last part, going to this, back to this slide here, okay? This last event that we're going to discuss is going to be the sarcomere crossbridge cycling. All right, we just talked about all this stuff here, trying to get the calcium out. Now, the final part, all right, the third event in skeletal muscle contraction is going to be, all right, the crossbridge cycling. Okay, so to start it off, we need calcium, which is going to bind to one of our regulatory proteins, troponin, which sits on top of the tropomyosin. And once that occurs, tropomyosin slides off, like you saw in the video, that rope, it slides off, all right, of the myosin binding sites, okay, on the actin there, so myosin can bind. So you can see these little black dots, those are the myosin binding sites, all right, where the tropomyosin was blocking it before. So once calcium sits on there, it changes the configuration, causes the tropomyosin to slide off, and now it exposes these binding sites for our myosin head to attach itself. Okay? So we're going to go through a couple of the steps here in a moment. All right, let me just pop this slide up here for you. Okay? And that basically explains to you what I just said. Okay? Keep in mind that calcium binds to the troponin. And it's the troponin that moves the tropomyosin off of the myosin binding sites. Okay, so troponin and tropomyosin are considered to be regulatory proteins. Okay, so to get those regulatory proteins the heck out of there, we need calcium. Okay, so the next couple of minutes, we're going to start talking about the crossbridge cycling. There's 4, steps to the crossbridge cycling. We're going to go through those. So what I'm going to do is I'm going to talk about one step, and I'm going to show you a picture, and then I'll move on to the next step so you can kind of get things in your head. I like to do it this way. Okay, so first step, okay, and these are repeating steps if we're going to keep contracting, all right, or they obviously won't repeat once we go into relaxation, okay? So the first step is going to be the actual formation of the crossbridge, meaning, all right, myosin is going to attach itself, all right, to the actin, okay? So to understand how this whole process works, all right, a couple things you need to understand first of all is one, that the myosin is in the ready position, or what we consider the cocked position. All right, so once it attaches to the actin, all right, then we can actually, in the next step, undergo the power stroke. So it's like if you've ever watched Westerns or the cowboys use a revolver in order to fire the gun, they have to cock the hammer back. All right, so that's what we're seeing here. All right, I'm going to come right back to this slide here. All right, so our first, I gotta zoom in. All right, here's our crossbridge formation. So the myosin head will extend across because we've exposed the myosin binding site. The myosin head extends across. It's in the cocked position here, all right, and it just attaches itself. That's it. That's the first step. Pretty easy, right, so far, okay? Now for the next step, all right, is the power stroke. This is when we are going to start the actual contraction process. All right, so at this point here, the thick filament is going to pull, all right, the thin filament past itself. All right, so we call it the sliding filament theory. All right, but it's called several different things. Okay, crossbridge cycling, sliding filament theory. Point being is we're moving 2 protein structures past one another. All right, so the thin filament will get pulled by the thick filament. Now, it's important that you understand this here. All right, ADP and inorganic phosphate are released during this process. Okay, ADP and inorganic phosphate are released during this process, which means we have a free binding site now for another ATP molecule to attach itself to. So we look here, zoom in again. On our crossbridge formation, if you look here, the myosin head, it has ADP and an inorganic phosphate attached onto this binding site here. Okay, this is all occurring during the crossbridge formation. Now, when that myosin head pulls the thin filament past itself, as it's doing that, the ADP and the inorganic phosphate pop off. So now it's got an exposed binding site. Okay? So now we're going to see ATP come into play here in a moment. Okay? So it's free. So this is the power stroke. We pulled the thin filament past the thick filament. We're starting to contract the muscle. As that happens, the ADP and the inorganic phosphate fly off that binding site. So the third step here, okay, now we're going to see ATP attach itself back onto the myosin head. Okay? When that happens, all right, this is when we see a release, all right, of the myosin head from the actin. I told you, all right, where a lot of folks will think that the ATP actually causes the power stroke. No, the ATP, when it binds on to the myosin head, causes the myosin head to pop off or release itself from the actin. And in doing so, okay, we move into the fourth step here. In doing so, that ATP splits into ADP and that inorganic phosphate. The enzyme, this is important, you want to know the name of this enzyme, it's easy. ATPase, all right, ASE is an enzyme, okay, so we name it ATPase, okay. This enzyme, all right, it lives on the myosin head. That's its home, okay? And we're going to learn that there's different variants of ATPase, okay, that allow for this process to be quicker or slower, okay? Fast variant, slow variant, okay? But it's this enzyme that is going to split the ATP into ADP and inorganic phosphate. And by doing so, it resets the myosin head. It cocks it. Okay, cocks it back. All right, so let me show you the picture and I'll come right back to this slide. Okay, crossbridge formation, we've attached, we have ADP and inorganic phosphate. All right, then we start to undergo that power stroke there, that pulling action. As that's occurring, the ADP and the inorganic phosphate pop off of the binding site there, all right, thus exposing the myosin head binding site for ATP. ATP moves onto the head, causes the myosin head to disengage, all right, it releases the myosin head, all right, from the actin, and then that enzyme, ATPase, will split ATP into ADP and the inorganic phosphate, and that process, all right, causes the myosin head to recock itself, all right, or reset, thereby allowing for another contraction, another pass, all right, or we go into relaxation. Nothing happens, okay? Yes, ma'am.
So the ADP and inorganic phosphate are released in the power stroke and when it resets?
In the power stroke, that's what, well, yeah, they're going to pop off, okay? But when it resets, they're still attached.
Okay.
They're still attached. This is not the best drawing here. It should show it right on there. All right, but they're still attached. Yep, they're attached here, and they're attached here during the crossbridge formation. They only pop off during the power stroke. Okay? Yep. All right, so let me go back to this slide here in case you didn't get all the information there. All right, so that's crossbridge cycling. All right, that is now, again, and we're going to talk about here in a second, that's just one crossbridge formation. Okay? So it doesn't mean that your muscle contracts all the way during that whole period. We're just starting to engage some of these actin and thick and thin filaments, all right, and we're starting to undergo some sort of contraction. Okay, it could be a weak contraction, all right. We're going to talk about the differences here in a second. Okay, so keep in mind, all right, in order to release that myosin head, you need ATP. That's why you need ATP and calcium for a muscle contraction. If you don't have ATP, you can't release the myosin head from the actin binding site. Rigor mortis, anybody? Okay, rigor mortis. All right, that's what rigor mortis is. Then eventually those proteins degrade anyways. You know that rigor mortis is not a permanent thing. You won't always be stiff like that. It's only for a certain period of time. Then the proteins start to degrade and then they break down anyways and it releases, everything just falls apart and the muscles go loose again. Okay? And calcium, we need calcium to expose the myosin binding site on the actin. Okay? So as long as you have ATP and calcium, there's a good chance you're going to have a muscular contraction here. Okay? So that's why, all right, those two things are present, we can keep crossbridge cycling all day long. Okay? And so during this time, as we undergo the crossbridge cycling, our Z discs will move closer together. All right? The H zone and the I band, all right, will start to narrow or completely disappear if we hit maximal muscle contraction. Okay, but they'll definitely narrow. I'll show you on the next slide here. But an important concept to understand is our thick and thin filaments never change length. They're always the same length. Okay, always. Okay, those are a couple concepts that you want to keep in mind. All right, so this next slide here shows you, okay, here's our kind of relaxed biceps brachii muscle. I think we all know that, okay? So you can see here, all right, Z disc to Z disc, M line in the middle with our myosin anchored to it. All right, here's your H zone. That's the zone that starts to decrease and narrow in the I band. That decreases and narrows. All right, possibly disappearing completely depending if we have muscle, complete muscle contraction. So here we are fully contracting our muscle. And you can see that pretty much the H zone and the I band are gone, fully contracted. But we brought our two Z discs closer together. The distance from here to here is a lot less than from here to here. Okay? And you can see it on our nice electron microscope here. Make sense? Not too bad? Okay. A couple questions for you then. What is the function of calcium skeletal muscle contraction? Basically, to remove the troponin, tropomyosin complex from the binding sites on the actin. That's it. Okay? And skeletal muscle. Okay, calcium plays its own role in the neuron function. All right? We need calcium to release the synaptic vesicles. All right, that neurotransmitter. So you can see the importance of calcium in your diet, in your life. Aside from strong teeth and bones, helps with neuron excitation and with muscle contraction. Then when you take 211, you get to learn about its role in blood clotting. So what causes the release of the myosin head from actin? There you go. There's our buddy, ATP. Boom! ATP causes the release. What resets it? The enzyme, ATPase. So when ATPase goes to work on ATP, it splits it into ADP and the inorganic phosphate. All right, so this next slide's got a lot of stuff on it. I wanted to put it near the end here, okay, because now what goes up must come down. So we're going to talk about, and you can probably figure it out on your own, what can cause relaxation, all right, of a muscle contraction. All right, how do we relax the muscle? Okay, there's a couple different places along that whole chain of events that we talked about. One, how about if we prevent the release of the neurotransmitter? All right, let's get rid of acetylcholine. Let's stop releasing that at the neuromuscular junction. Okay, now we never get an EPP, then we never get a muscle action potential, and calcium never gets released from the sarcoplasmic reticulum. Okay? So we can see this guy right here, acetylcholine esterase. We talked about that at the end of chapter 12, all right, about how we start to break down neurotransmitters. Acetylcholine esterase will break down acetylcholine into acetate into choline. All right? And then the synaptic knob can reuptake the ingredients and then build it again inside the cell. Okay? If we close off the chemically gated channels in the motor end plate, we can stop an EPP from being created. All right? Then, if that occurs, then we will not have an action potential. Okay? If we don't have an action potential, then the calcium channels close. Calcium will no longer be released, all right, from the sarcoplasmic reticulum terminal cisterna, and our calcium pumps will pump that calcium from the sarcoplasm back into our sarcoplasmic reticulum terminal cisterna, right? Thereby allowing troponin and tropomyosin to slide back over those myosin binding sites, stopping the contraction, all right? And then muscle can then go back to its original position due to this characteristic, one of the many characteristics of muscles. I hate that. Elasticity. Remember some of the characteristics? Conduction, all right, excitability, okay? All those fun characteristics that we went over in the beginning of this chapter, okay? So, I mean, if you think about it, if you kind of walk through, so when you're looking at these steps here on this slide, all right, you can kind of figure it out. I know it seems like a lot, all right? We went over a lot. but if you just kind of think it through, you'll be able to figure it out. All right, if we can prevent calcium from being released, we'll relax the muscle. If we can somehow, I don't recommend it, deplete all the ATP stores, all right, therefore you can stop muscle contraction. Okay? All right, so how do acetylcholine esterase and calcium pumps function in the relaxation of muscle? Basically just kind of what I said, all right? That enzyme is going to cause acetylcholine here, all right, to disconnect through hydrolysis from the receptor. We break it down into acetate and choline, okay? And therefore it will no longer stimulate that muscle fiber, and the calcium pumps then are going to move calcium back into the sarcoplasmic reticulum. And then that troponin triple myosin then close over the binding sites on actin. Okay, so we're going to not really change topics. We're going to stay on skeletal muscle. But now we're going to learn about how are we able to supply skeletal muscle with energy. Okay, and so there's a couple different ways that we're going to do that. But we're going to talk about how we get energy to, all right, these cells here. Okay, so keep in mind, all right, there's two tissues, organs, that actually contain stored glucose. Because remember, glucose, we break down glucose, and that gives us ATP through metabolism. Okay? Well, there's two tissues that actually have stored glucose, which is called glycogen. Okay? The liver and skeletal muscle. Okay? More on that in a moment, but I want you to keep that in mind. So, when we're dealing with energy, I want you to understand ATP, all right, is made by the cell membrane, okay, and it's also made by, or the majority of it, by the mitochondria. So ATP just gets pumped out into the cytoplasm and it just floats around out there. So it's on hand for a lot of things, okay? So we're going to have ATP throughout the cell. But unfortunately, if you're going to be exerting yourself, all right, strenuously, you only have about 5 seconds worth, okay, that are stored right there in the cytoplasm. Okay, that's a problem. All right, but no problem. We can get more ATP, all right, from this enzyme here called myokinase. Okay, myo is muscle. Kinase is an enzyme that likes to put phosphate onto things. It'll stick phosphate onto things. So in this case, it's going to take phosphate from one ADP, and it's going to stick it onto another molecule of ADP to make an ATP. Dr. Kaz, what are you saying to me? Let me show you. I'll come back to that slide. So here we are. We got two ADP molecules. All right, that means we have a total of 4 inorganic phosphates between the two of them. Adenosine diphosphate, that's what it means. Okay, so this awesome enzyme is pretty much going to pluck off, all right, one of the phosphates off of, we'll say this one. It's going to pluck this phosphate off of this ADP molecule and stick it onto this one. So we're left with ATP, whoops, come on, ATP and AMP. So we now have an ATP molecule, okay? So that's what the kinase does. It's just going to take a phosphate, all right, and pluck it off of one molecule and stick it onto another. Okay, so that's one way. That's cool. That's cool. All right, I can go with that. But we got to have more ways to make some ATP here. All right, especially if you're running a marathon, you can only do this for so much. So we have some more ways. We're going to talk about these. This awesome guy, creatine phosphate. Well, we've heard about glycolysis before because I talked about it earlier in chapter, I don't remember, 2 or 4. All right, so bad I can't remember anymore. But glycolysis is when we start to release glucose from its storage form of glycogen. Remember, glycogen is just a whole bunch of glucose molecules strung together. So we're going to just start chopping those pieces off. All right, so here's a glucose molecule attached to another glucose molecule attached to another glucose molecule. All right, and so through the process of glycolysis, I'm just chopping them off and then releasing glucose into the bloodstream. Okay? And then finally, aerobic cellular respiration. All right, for that one, mitochondria. Mitochondria. Powerhouse of the cell. I won't spell it out. All right, mitochondria. Okay, so let's start off with the first one. All right. So again, this is initially what I just talked about. All right, that's one of the ways that we can make ATP very fast. Okay, but eventually that's not going to cut it. All right, no problem. We got another process. All right, if we need to rapidly make some ATP, okay, it's only going to work for a short period of time. All right, but we've got this creatine phosphate. All right, so again, phosphate. Ooh, yeah, phosphate. We need phosphate. We need phosphate to make ATP. So what happens here is we've got this enzyme, it's called Cretan Kinase. There's that kinase word again. Oh, that's an enzyme that takes phosphate off of one molecule and sticks it onto another. Okay, so what's going to happen here? Well, we're going to take the phosphate off of this creatine phosphate, and we're going to stick it on an ADP, and guess what? Oh, we got ATP. How do you like that? So that's cool. This will give us about 10 to 15 more seconds. That's not going to do us any good in a marathon. Maybe for the first 100 yards. No, I'd say a little bit longer than that. All right, less than a quarter of a mile, but it's still better than nothing, right? Okay? So I'm going to come back to this slide here. That's what this is, right here. Here's the creatine kinase. So we have our ADP, all right? And then we got our creatine phosphate here. So we're going to take the phosphate off of the creatine phosphate and stick it on to the ADP. That'll give us ATP and creatine. Cool, right? Bodybuilders figured this out a while ago. They take this stuff called creatine phosphate. Oh, wait, I just said that. Yeah, they take that stuff because it helps them to recycle, all right, ATP when they're trying to do these quick bursts of workouts. Okay? Problem is, it gives you bad diarrhea. That's true, honest truth. Okay? So this is good. All right, 10 to 15 seconds. All right, okay, all right, so that's good. But now let's talk about a little bit longer. I need more, you know. I want to go for a bike ride for at least an hour. So I need energy for that bike ride. Okay. So let's take you to the next method of creating energy, glycolysis. I love it, okay? Now, unfortunately, glycolysis is anaerobic, so that means it doesn't require oxygen, okay? Well, that sucks because oxygen is great because it allows us to get more ATP out of one molecule of glucose, but hey, you know what? It's better than nothing. So we'll go with the whole anaerobic situation here. So we have one molecule of glucose, and so it's going to undergo glycolysis, okay? And so that glycolysis, all right, is going to break down glucose into pyruvate. So one molecule of glucose is going to yield us 2 pyruvate molecules. And those 2 pyruvate molecules without oxygen will yield us 2 ATP. That's not bad, but there's a way better way, all right, to get more ATP. And we're going to talk about that in a second. And I have to correct myself. My brain's going a million miles a minute here, okay? I'm going to correct myself. I just realized something. Glycolysis is the breaking down of glucose into pyruvate. Here, I misspoke and said on this slide that glycolysis is breaking down glycogen, okay? That's glycogenolysis. I'm putting that out there. I'm going to spell that. All right. So what I misspoke about, get rid of that, is glycogenolysis. Glycogenolysis is when we have a glycogen molecule, so it has all these strung along glucose monomers, all right, and then we just start cleaving the monomers off. Okay, that's glycogenolysis. That looks awful. I apologize about that. I misled you. Glycolysis is this, when we start to metabolize and break down glucose, all right, into pyruvate and then eventually into ATP. Okay, I just want to correct myself. I'm embarrassed. I shouldn't have missed that. Okay, so Glycolysis occurs in the cytosol. Okay? So this works for a little while, depending on how much glucose is on hand. All right? If your blood sugar levels are pretty decent, you know, you'll burn through that. Okay? But now we've got to figure out a way to get some long-term energy. And this is it. This is the swan song. This is the beautiful, beautiful metabolic process of getting a lot of ATP, all right, a lot of ATP from glucose, okay? But you need cellular oxygen. We need oxygen for this. And the only way that we can have this metabolic process occur, all right, is with oxygen, and it has to be in a special place, and that special place is club mitochondria. Okay, and oxygen is that friend that's going to get you into that club. All right, so oxygen is going to be pyruvate's friend. Okay, so glucose comes into the cell, all right, undergoes glycolysis. We break it down into 2 pyruvate molecules, and if oxygen is present, then that oxygen is going to take that pyruvate and bring it into the club, bring it into mitochondria. Okay? And when it enters into mitochondria, it's going to go through a ton of chemical reactions. All right? Have you ever heard of the Krebs cycle or the TCA cycle, tricyclic cycle? All right? That's this process that occurs. Tons of steps. Tons of steps. You get to learn about that in 211. All right? But basically, we're going to wind up near the end here, those chemical processes with these guys, these coenzymes, these structures here. But essentially, NADH and FADH2 is going to be taken to this place called the electron transport chain, or ETC. Okay? And it's going to cause these energy bonds to be cashed out as 30 ATP. Okay? That process is called oxidative phosphorylation. All right? I don't expect you to know and memorize everything, but the point is, what I want you to know is this requires oxygen. It's going to occur in the mitochondria, okay? And we're going to yield 30 ATP, all right, from those pyruvate molecules. Okay? And our waste product for all of this, carbon dioxide. All right, well, that's cool, carbon dioxide, because the cell is just going to kick that back out into the bloodstream, and then you're going to exhale that off. And there it goes, out into the air. Okay? So, we see that occurring. Remember, pyruvate, that's what glucose, all right, gets converted to or broken down to. Okay? And if there's no oxygen, then that gets converted into the ATP molecules. I should say it gets converted into, but we are going to generate energy from that to give us the ATP molecules. Now, triglycerides, fats, remember fatty acids? Okay, fatty acids are made up of a glycerol group with some fatty acid chains. All right, well, these triglycerides here are the preferential fuel choice. Okay, fats are the best. Preferred fuel of choice. All right, again, we need oxygen for that. Okay, so we can get more ATP from these triglycerides, especially if they have, the longer the fatty acid chains on those triglycerides, instead of having 10, all right, 200 or 100, all right, the longer the chain, the more ATP we get. Okay, and skeletal muscle loves that stuff. Ideally, aerobic respiration is the way to go. Okay? Aerobic respiration is the way to go. Because also, when you're undergoing aerobic respiration, on average, the fuel that you will be burning will be 50% carbohydrates and 50% fat with oxygen. Once you engage in anaerobic respiration, you're going about a 20-80 ratio, well, I should say ratio percentage. All right, 20% will be fats and 80% will be carbohydrates. Okay? So that's why it's ideal, especially if you want to, if you prefer to burn off more fat than carbohydrate, then you want to roll with an aerobic type of metabolism here. So this slide is just showing you all of these awesome processes at work here. Okay? So you can see the first two processes here, all right, when we're talking about Myokinase, Creatine Kinase, all right, that does not require oxygen for those purposes. When we get into glycolysis, again, you don't need oxygen. You'll get some ATP, all right, but you're only going to get about 2 per glucose molecule. Here now, we're rolling into cellular respiration. Now we're going to deal with oxygen that gets us into the mitochondria so we can enter into the citric acid cycle, or the TCA cycle, Krebs cycle, whatever you want to call it. All right, and then we can start to generate way more ATP. The nice thing is, too, look at all this stuff that we can throw into this citric acid cycle, into the Krebs cycle. You can use fatty acids, all right, from triglyceride groups. We can even use some proteins when we're dealing with gluconeogenesis. There's a lot of stuff, okay, because the proteins break down into amino acids. Let's throw some amino acids in there. Have some fun with that, okay? So again, these are just a bunch of the different ways that your muscles are actually going to engage in some of the processes there, depending on what fuel source and what type of metabolic demands you placed on your body. Does everybody hate having sore muscles? You ever done an activity for whatever? Like, I once walked really fast for a really long time and I had shin splints the next day. All right? Shin splints are a different animal, but my calves were sore horribly from that because I was doing a lot of uphill walking. A lot of that was from this guy here, lactic acid formation. Okay? Well, guess what? In some situations, all right, if you don't engage in, all right, aerobic cellular respiration, if there's no oxygen, you are going to start to accumulate lactate. Okay? So if we have very low or no oxygen, pyruvate can get turned into, so here's the glucose molecule. At the end of glycolysis, we have pyruvate. And so pyruvate has a couple different options. It can go into the mitochondria, all right, and go into that Krebs cycle and get turned into 30 ATP. Okay? But if there's no oxygen, then it can go and be shunted over here and get turned into lactate. Okay? Now, again, the skeletal muscle cell, can use lactate as a fuel source. Okay? It can. All right? Because what will happen is either the skeletal muscle cell, all right, or, well, we're not really talking about the cardiac muscle cell, it can too, but the liver, all right, can change lactic acid or lactate, all right, into glucose through the process of gluconeogenesis. How cool is that? Okay? So the skeletal muscle fiber, all right, can use the lactate, or that lactate goes into the bloodstream, travels to the liver, and then the liver then starts to convert that lactate into glucose through gluconeogenesis. Glucose, neo is new, making new glucose from lactate. And then it goes back to the skeletal muscle, and the skeletal muscle then can break it down into pyruvate again. And hopefully maybe this time there's enough oxygen and it can convert it into, all right, 30 ATP. That's cool. All right, or it can get converted back into lactate, and then you just wasted your time. All right, but you still got 2 ATP out of it, going from glucose to pyruvate. A lot of this will make more sense when you get into metabolism. I keep saying stuff like that, but you learn that. You'll learn it in the course. You just won't learn it now because we have many other things to talk about. All right. Okay. So when does this stuff occur? Okay. So for short bursts of energy, about 10 seconds. So perfect example for a 50-meter sprint, we are going to use that phosphate transfer system. What's that? That's this right here. That's the phosphate transfer system. Myokinase, Creatine Kinase. If you look at the word phosphate transfer system, remember what I told you, we're going to transfer a phosphate from one thing to another. That's what that is, okay, in case you weren't sure. Okay, so that's going to occur primarily for short bursts of energy. All right, now, for a little bit longer burst, but still short, less than a minute, about a 400-meter sprint, that's once around the track. All right, we're going to start off by using the phosphate transfer system, but like I said, that's only good for 10 seconds. Well, what do you do after that? No problem, glycolysis. We got glycolysis. Okay, so all that blood sugar that's floating around, we're going to break that down, bang, all right, and we're going to make ATP with it. Okay, that's cool, but what about a longer run? Maybe a mile run. If you're in a cross country, you might do a 5K or something. Okay, so more than a minute, we're going to use a combination of all three, obviously. Okay, start off with the phosphate transfer system, undergo glycolysis, but ideally, we want the aerobic process. We want to get mitochondria involved. Okay, so the aerobic cellular respiration process. All right, that's what we want. That's why training is so important, because you're not only having to train the muscles for the actual activity, but you got to train your body to utilize the right fuel source at the right time. I'm not big into running, but a lot of my patients are, and so they drink this goo stuff, and it's just loaded with a good fuel source. It's liquidy, so your digestive system can break it down. Keep in mind, when you're running and doing certain like marathon activities, your digestive system is not a high priority system to be operating. All right, so blood gets shunted away. It's the fight or flight. We'll talk about this later on, the sympathetic versus parasympathetic. All right, so it's not high priority, but you still need to get energy from something, so they'll drink this goo stuff, and it helps with that. All right, so ideally we want patients to engage, or people to engage in aerobic exercise, okay? This aerobic exercise, all right, you're going to hit a target heart rate, okay, which is going to be above just an elevated baseline heart rate, all right? But what it does is this. Enhanced oxygen delivery to all the cellular tissues that need it. Okay, especially if you're going to be doing long-term exercise at a higher intensity. Aerobic exercise is the way to go, and it goes back to what I said before. All right, that 50-50 percentage there, 50% carbohydrate breakdown, 50% fat breakdown. Okay, for the fuel source. All right, that's ideal. So that's what this picture here just shows us, the utilization of the energy processes, all right, that short burst, all right, the phosphate transfer system, all right, once around the track, that's going to be glycolysis and the phosphate transfer system. Then that 1,500 meters, okay, I think that's 4 times around the track, okay, I never ran track. That's going to be primarily the aerobic cellular respiration. All right, 2 more slides. Additional ATP is made immediately available in muscle tissue through which unique phosphate-containing molecule? Creatine phosphate. And then what are the 3 various means for the 1,500-meter race? All right, it's going to be the phosphagen system or the phosphate transfer system, anaerobic cellular respiration, which is glycolysis, and then aerobic cellular respiration. That will be primarily the one used, that means primarily, all right, during that period of time, as long as oxygen is present. We need oxygen for that to occur. All right, great job, y'all. Let's take a break and rest our minds, and then we'll continue on with this marathon of muscle.